Hi, hi to the series of TEFLY, again, Scien-TEFLY, not scientifically. Today we'll talk about TOC also. We started about it in previous video. Today we'll take actual introduction for TOC, total organic carbon analysis, but as a practical introduction more than theoretical. We will make like skimming, quick skimming, hopefully to be useful. So today we'll talk about TOC quick introduction lab. This is we are not talking about this dark, okay? Okay, but this because TEFLY, TEFLY, we'll name it TEFLY for as a fun. Okay, okay. So can you expect from your suppose you are at least started working in the field of laboratories. So this video is considered to be specific, but also we are trying together to make it simple. So we are talking about TOC. So in lab we analyze many parameters like starting from simple analysis like pH, conductivity, TDS, salts dissolved in water or any samples, liquid samples, like in pharmaceuticals also water for analysis. So we will talk about TOC, where we can find TOC. I'm trying to, as we said, TEFLY. Okay, we'll talk about organic carbon. Okay. So organic carbon, where it can be found, for example, in the sea, in the agricultural, in plants, or I missed the name of coral reef, or algae. Okay. So this is the sources of organic carbon. Okay, we'll continue again. And of course, we when we do any analysis in the laboratories, environmental laboratories, pharmaceutical laboratories, sorry for this. What is laboratories? What is this? Okay, laboratories, pharmaceutical, environmental, water, industrial laboratories. So analysis takes many forms. We use instruments for this, and hopefully planning to make specific video for references. I use it generally, modestly of course, and I'm trying to do something fruitful together. Wanna be fruitful with you. Okay. So before we talk about instruments, we think about the theory. First thing that comes in mind, how this instrument measured a specific parameter, for example TOC, how this measures this TOC. So when we should take a look to TOC. Do you have idea about how TOC is measured? Titration, carbon titrated? No, something like that. We may use type of radiation like spectrum, near. Okay, we'll see here. UV. Okay. I think till now we are TEFLY, trying to be illustrations and like TEFL. Okay. UV. Produce. This numbers is not accurate, and I'm doing this video based on experience, hand-on experience and previous information. Not accurate information from references right now. Of course, read before, but not right now. So numbers may be okay, not accurate, so accurate, but in this range. UV produced two wave numbers, 250 and 180 nanometer. So these two wave numbers is responsible for producing OH radical, and this comes through water itself. From UV radiation irradiate water, so produce OH from water, And also another way, so we use with UV some chemical called pair sulfate. Okay, so pair sulfate also is a way. There's equations we'll talk about in next videos. Finally, we get OH radical. So this OH radical is the actual player who interact with carbon, organic carbon, whatever the source. So produce at the end CO2. This is a core idea of TOC, total organic carbon analysis. So this stage of analysis itself, or theory of the instrument. Okay, there are many types, as I said, for TOC analyzer, famous, and we'll talk now about detectors. The main difference usually in this step, analysis step, some instruments I explained UV, but some instruments use the combustion. Actually, I think this is main two ways. I don't know. I'm not sure about if there are new ways or older ways that not are used now. But the main UV and combustion for analysis. This is analysis stage. Analysis. We will talk now about detection stage. But for analysis, we explained UV. Combustion is similar but depends on high temperature, but the idea of getting OH radical. Okay, we'll get now into detection. Sorry if I'm talking about, I'm doing like scanning, as I said. So maybe a lot of informations, but I'm trying to make it simple as I can. Hopefully I can. So we'll transfer into detection stage. One moment. For detection stage, we are now talking about detection. Okay. Sorry, should talk together first to try to concentrate. Detection stage, can you expect? How can we detect? We detect by for example, GC, we have many detectors. FID, flame, ionizations, type of flame, which detect. I'm not so specialized in GC, but as I know, GC detect by flame for specific compounds using flames. Of course, GC have thermal program like that, but I'm trying to make something close. Can NTUC detect by flame? We said in analysis stage we make UV or combustion. So detection, the actual measuring of carbon comes through measuring the released CO2 through what? Can we measure carbon dioxide gas, which represents to us carbon at the end, of course. So organic carbon, we measure it as in the form of CO2. So how can we measure this CO2? CO2 is measured actually like, can you guess, right? Type of spectrum also? Maybe, yeah. Okay. This right, this spectrum, and which is called, let's reveal part, NDIR, non-dispersive infrared detector. Actually, that's right. Who said this? Okay, excellent. NDIR, non-dispersive infrared. This is measuring the TOC, actually CO2, which is released, measures the CO2 in the form of NDIR detector. Not in the form, sorry, by using NDIR as a detector. So this is the detector. It takes this shape, integrated inside the TOC analyzer itself. Here, this is TOC, and the detector take a part of the analyzer. This is type. But as we said in the analysis stage, we have UV, you have combustion. Also in detection, we have actual two types, I think, mainly. Yeah, NDR and the other type. Can you expect? We said no flame. Spectrum already said what, like MS, electron beam. It's similar to spectrum, no. actually something different, and this actually unique feature of TOC. Actually, I like this instrument actually because it's contain technology and simplicity in the same time. Actually, real. So the other way is conductivity cell, something like pH, conductivity TS. This is actually, yes, detector conductivity cell. And from experience, I'm not recommending type over type, as both are great in detection, but I think conductivity cell is very accurate for maybe low concentrations, although NDR also measure in low concentrations, but conductivity cell actually is very innovative technique, actually, yes. So here CO2 change the conductivity of DI water. There are a tank and types that use of TOC analyzers that use conductivity cell as a detector. These instruments actually contains container or yes, inside tank, small tank that of DI water inside the instruments that is already inside the instruments, and the measure maybe regenerate from time to time. Okay, but this water, when CO2 is released from our samples due to organic carbon, of course, the CO2 changes the conductivity of this DI water, deionized water, which typically stable, changes over the introduction of sample and analysis and detection step. This change and directly measured by the instruments as a reading to concentration. There's also, but I don't go to more details. Some instruments measure TOC directly, some indirectly by subtracting inorganic carbon from total carbon and measuring TOC carbon. This is will be in other videos. I think this enough as not became quick it became longer, somewhat introduction. So I think it is enough for now. Hoping that will be fruitful, and see you for other videos soon, hopefully together. Thank you very much. I will make a quick brief. One moment. So as we see here, TOC is measured by either by the measuring stage by UV persulfate or combustion, and the evolved CO2 is measured through two techniques of detection: NDIR, non-dispersive infrared, or conductivity cell. This is a brief about what we said. Thank you and hope to see you soon. Thank you.